Diagnosing an Ancient Form of Pagets Using Modern Methods. Many of you who are members of the Pagets Association and who receive the magazine monthly will have perhaps followed some of this story. It's something that's been fairly high profile, or certainly a focus for the Pagets Association for the past year or two. And this is work, or includes work from a number of groups across the UK and actually internationally as well. So this has been a large project which has evolved, and I'll tell you about some of the elements of that work, and particularly a focus of the work that we've done here in Nottingham as part of the Pagets Association Centre of Excellence. So I'm a basic scientist. Many of the speakers you've heard today are clinicians, so they deal with patients, they meet people. We deal with proteins and we deal with pipettes. So we deal with experiments that help inform and understand the mechanisms that underlie diseases. We were a lab that researched contemporary Paget's disease, so modern Paget's disease, the type that we've spoken about today, the condition that affects individuals in this room, until about 3 years ago, and by a series of very strange coincidences where Diana was walking a dog one day and she met someone. It's a long tale which I'll maybe share with you later as well. But essentially we were put in touch with a museum in the northwest of England near Runcorn called Norton Priory Museum, and many of you will have read about the unusual tale of the mystery of Norton Priory and the connection with Paget's disease and maybe an ancient form of Paget's disease. And so that's the story that I'll elaborate on now. Diana mentioned we had a patient day a week ago today, a really successful day, actually, our members' day, should I say, at Norton Priory. So those of you who don't know, Norton Priory is close to Runcorn. It's probably about an hour and 40-minute drive from here. And it's the largest monastic excavation in Europe. So it was the site of a medieval priory which was founded in 1134 and dissolved in about 1523, I think. And on the site of Norton Priory then there is a museum, a really wonderful museum. It was recently refurbished with funding from the Heritage Lottery Fund, a really beautiful place to visit. And I would encourage any of you who haven't been and perhaps are in the area or are able to travel there to perhaps think about spending some time there, because it's a really special, peaceful place and a fantastic museum for the Northwest as well. It really is a flagship. So Norton Priory was this medieval priory, and on the site then there were burials of over 130 individuals who were associated with the priory back in late medieval times, the 12th through 15th century. And many of these individuals were high status individuals. They were canons, they were noblemen, they were crusaders, they were knights, and they went to the priory, or they were associated with the priory because they made vast donations of cash, because this was one way that afforded you a better route or a faster route that way after you pass away rather than that way. So that was the thinking at the time. So over the past 20 years or so, something like 130 skeletons, human skeletons, have been unearthed at Norton Priory and very carefully collated, respectfully treated. And on the right-hand side is just an image from the inside of the Norton Priory Museum, and as I say, it really is a beautiful place. So I would urge you, it's not often I recommend Runcorn for a weekend away, but in this case maybe. So what's the connection with Paget's then, for those of you who perhaps don't know some of the story? So I'll introduce to you an individual. This is a facial reconstruction of one of those 130 individuals who lived at Norton Priory in late medieval times. So this was, we believe, a gentleman called William Dutton, who was a medieval canon at the priory. So this is what we think he would have looked at, and one of the partners in the research has performed this reconstruction, and he's photographed, or his reconstruction then is photographed in the grounds of the priory. So he probably looked something like this. And in our research connection with Norton Priory, we've been drawing together lots of expertise to really try and enrich the narratives surrounding the life stories of individuals at the Priory. And at the bottom of this slide you see why the Paget's Association is interested, because one of the things we've learnt from the analyses is that William and many of the other individuals in the Norton Priory collection, many of the other 130 individuals, we believe was affected by an ancient and very unusual form of Paget's disease. So we have insights, if you will, into the natural history of Paget's disease, how it's evolved over hundreds of years. So the type of work we and our collaborators have been doing have helped populate the left-hand side of this slide what we know. So, for example, from simple radiocarbon dating and analysis of architecture, we know that William died probably in the late 14th century, so late medieval. He lived to a reasonable age actually, so from analysing skeletal features and the traits of his skeleton, he was probably in his late 40s, which wasn't too bad actually at the time. But the thing that interests us and the thing that led to my own research group and the Paget's Association partnering with Norton Priory was this observation that his skeleton seemed to be affected with an unusual bone disease. And this unusual bone disease affected about 3/4 of his skeleton. So on the left-hand side is a cartoon representation of the ancient skeleton of William, and the sites in yellow and red indicate the bones in his skeleton that showed changes that resemble Paget's disease. So extensive is not the word, you struggle to find somewhere that's not affected, I think. So the bottom line is really the contribution that research in the University of Nottingham and in my lab from my research group has done. And we believe that we've confirmed that although his disease was unusual, this was a form of ancient Paget's disease. So Paget's disease in the northwest in late medieval times was very different to Paget's disease today. And the way in which we've done this is through analysing proteins that have been preserved in William's bones for 800 or 900 years of age. So that's the story, again, that I'll put some more detail onto. So I said this is a Paget's-like disorder, and again, over the past 4 or 5 years, a very detailed analysis or reanalysis of skeletons in the Norton Priory collection, so those 130 skeletons with the Paget's-like changes, has been performed, and much of this work has been performed at Liverpool John Moores University, once again partly supported by the Paget's Association. The sort of changes you typically see in the remains of those affected are shown at the top. So this is a femur of an individual in the Norton Priory collection with Paget's-like changes. And again, you don't need to be a pathologist or a bone pathologist to see that this part of the femur is smooth, this part of the femur is normal, but this end of the femur is very different. It's enlarged. It shows visually changes that are consistent with Paget's disease. And if we use imaging approaches, so the bottom is an X-ray of that same femur. I think in fact this may have been William's femur. And once again, there are characteristic changes on the X-rays that resemble modern-day contemporary Paget's disease. So a Paget's-like disorder certainly, but the reason we kind of counter this with like disorder is because, as I've already alluded to, there are many, many differences in the individuals affected with this ancient bone disease compared to contemporary cases of Paget's disease. And so, of course, if it looks like a duck and swims like a duck and quacks like a duck, it almost certainly is a duck. My analogy is that you could say the Paget's-like disorder at Norton Priory has some features of a duck, but it's a mammal, it's a duck-billed platypus, if you will. And to emphasise really what those differences are, I've tried to tabulate on this slide. So it might be busy, but I'll just walk you through it. And I think it will become really apparent then that there's something very unusual about this ancient bone disease at Norton Priory. So you'll have heard today, modern, contemporary Paget's disease. We think the current incidence is in the region of 1% of individuals who are over 55. That number is dropping. So there are certainly changes in Paget's disease going on at the moment. Paget's seems to be becoming less common and also less severe, and maybe also presenting at a later age. But we're not far off probably with 1% of the population over 55. In the Norton Priory collection of those 130 skeletons, we think as many as 20% of the individuals had those characteristic changes, might be even higher than that. So certainly in the adult sample, many cases, not just William, but many other cases of Paget's-like disorder. And I already told you that William's example is that these skeletons, the affected skeletons at Norton, can have very extensive changes. So I showed William's representation of maybe 75% of his skeleton affected. And this contrasts with the things we've heard about today, this idea of monoostotic Paget's being common. Today, maybe only one or a few bones are typically affected in those who have Paget's. So a really big difference there. Age at onset as well. So the example of William, he'd lived to a reasonable age. He was in his late 40s. But many of the skeletons show Paget's-like changes that would be consistent with onset in young adulthood. So early 20s, possibly even younger as well. And we really don't see that today. It's highly unusual, I think, to see Paget's in individuals under 55 years of age. But the final kind of strange thing, the mystery, is despite these differences, despite the fact that Paget's was so extensive, complications, and the type of complications, again, we've already discussed, although reasonably common today, we talked about fractures, pseudo-fractures, bone pain, although obviously we'd be speculating about whether bone pain was significant back then, but things like bone deformities as well. You just do not see them in the Norton Priory collection. And of all of the skeletons analysed, there's just a single bone that I'm aware of that showed any evidence of deformity. So this is a real mystery. It seems to be an extensive disease, but somehow it doesn't seem to be severe, at least based on visual complications. And naturally, this led us to an awful lot of scratching our heads and debating at length, was this really Paget's disease, or was this some related or unrelated bone disease that had been completely missed actually in the archaeological record? And we grappled with this question for a long time, and then we became involved in research and we attempted to experimentally address this question. And the way we did this, again, it was ambitious, and I can remember at the outset thinking, this is never going to work. It's just way too difficult. So we knew a few years ago that actually proteins, so proteins are one of the molecules in our cells, can be remarkably stable. So probably 10 years ago, you would have assumed that proteins had a stability after we'd passed on of a few weeks or months or maybe years. But research started to show, maybe about 5 years ago, that some proteins, molecules within us, are remarkably stable. And it turns out the proteins that are stable are in mineralized tissues, such as bone, and other calcified tissues, such as teeth as well. So we knew that. And what we set out to do then was to ask this question in blue: could ancient bones hold what we termed a chemical memory about the associated disease or diseases that may have been affecting individuals from which those bones came. So our specific question was, could samples from the Norton Priory collection shed a light on a retrospective 800-year later diagnosis of the disease that was affecting those individuals? And the answer was remarkably yes, and I really did not expect this to come off. It was an enormous study really that involved tremendous hard work from people, many groups in many places, with lots of support both from the Paget Association and other funders as well, the Wellcome Trust, Michael Davey Foundation and a variety of other funders. But this is kind of the outcome, this is the headline, and this is a scientific paper that we, the research team, published earlier this year, which attracted a fair amount of media attention. So this described our work, and you'll see that there's work from Nottingham, but also from East Anglia, from Liverpool, from the museum, Leicester, Liverpool John Moores, Oxford, and so on. So many people contributed to this, and we were privileged to be able to coordinate the study. I'll point out that Barry Shaw, who was the lead on this work, is sat at the back. I heard him embarrassing me at lunchtime, so I can return the favour as well. But he did an awful lot of this work here as well. And Ana Navarro Martinez, so she's also one of the contributors, and she was a recipient of one of the Paget Association student research bursaries. So that had a major impact as well. As I said, we had media attention. We were in BBC History magazine, Current Archaeology. We even made it into the Daily Mail online. But essentially, we were able to diagnose an ancient form of Paget's disease with an awful lot of hard work from the people I've mentioned and also other individuals as well. And the way in which we did this is to come back to Nicole's work, being molecular, if you will. It's analysing molecules, but in this case, ancient molecules that had survived 800 years or more. So Nicole told us about DNA and genes and reminded us then that we've all got this blueprint of DNA in our chromosomes, or our chromosomes are made of DNA. And actually, as humans, we probably have about 20,000 different genes. Those 20,000 different genes make those RNA molecules that you've briefly heard about, and they go on and they make the proteins, the toolkit, the functional entities of cells. And in terms of complexity, we've probably got 100,000, maybe 1 million different proteins. So we're quite complicated individuals, actually. And my lab specialises in analysing proteins and learning information about proteins, and we're very interested about proteins and disease. So the sequestosome protein that Nicole mentioned is just one of those 100,000 or million different proteins. So if you think back to our hypothesis, we wondered whether samples of ancient remains might have this molecular memory buried in their proteins. And actually, remarkably, Barry and Anna and others then were able to take small samples of bone tissue from affected individuals at Norton Priory and to actually extract 700 or 800-year-old protein. And that's really what this schematic shows. The thing on the right-hand side that looks like a brown mess is a biochemical analysis, and to us that's really exciting. That tells us that there is ancient protein that survived in these samples for millennia. Millennia? No, for hundreds of years. I'll get my facts right. So this was the first stage. We're able to extract ancient protein. But what matters is whether we can use the information that's retained in that protein to try and diagnose the condition. And we can have a quiz if you like, but I'm sure most people in the room will know the movie this comes from. But this really kind of reflects to me how I felt when we got the data back from the first analysis of those ancient proteins. So I said there are 100,000, maybe 1 million proteins that we have. In bone, there's probably 10,000 or 100,000 different proteins. We knew we'd been able to extract ancient protein, and our question was, what are those proteins? And so there are various techniques one can use to identify different proteins, and that's essentially what we did in our experiments. So we catalogued, if you will, the ancient proteins that have been preserved in small samples of bone for hundreds of years. And so the title kind of gives it away. Of the tens of thousands of possible bone proteins that would have been there at the start, after 700 years, we were able to identify only 18 proteins that had survived. And you kind of know where you are when significant events happen in history. I can remember where I was when Donald Trump was elected. I can remember where I was when Nottingham Forest lost in the FA Cup final. I was in Wembley, 1991. And I can remember where I was when this data came back. The 18 proteins then are listed on the right-hand side, and again, don't worry about the complex scientific names, but I was in my kitchen. It was a Tuesday afternoon, and I was making a salad, and I wasn't focusing on making tea. I had my laptop set up in the kitchen, and I was checking my emails, and this data came through, and the list is shown on the right, and I read it, and to use a phrase, I nearly dropped my cucumber, because in that list of only 18 proteins that survived, one of them, shown in red, is the sequestosome protein, and this is the same protein that Nicole will be studying in her research. This is the protein that's intimately associated with Paget's disease. So if you will, this is a marker or a biomarker of Paget's disease. The other 17 proteins are things you would expect to see in ancient bone samples. So collagen is a very common bone protein. It's the most abundant and probably one of the most stable proteins that we know. So it wasn't a big surprise. And some of these other proteins, keratins and albumins, that they're always seen. But this sequestosome protein has never been reported in any ancient samples by other individuals. And so its identification alone was really good evidence that we're looking at an ancient form of Paget's disease. So it's different. It's a very different form of the disease. Phenotypically, it looks nothing like contemporary or modern Paget's disease. But we believe, based largely on our protein sequence analysis, that this is ancient Paget's disease. And there are other factors as well, and other molecular analyses that back up our proposals. So not only could we just detect the presence of this ancient protein, but remarkably, we could read much of the sequence of this ancient protein. Ancient DNA sequencing is something that people have done for many years, and it is possible to get information on genetic material, but sequencing ancient proteins is very different. But somehow this sequestosome protein not only survived, the Paget's associated protein not only survived, but it was intact and we could read its sequence. So this is the code, if you will, that is the sequence of human sequestosome protein, and anything in red is the ancient protein sequence that we were able to read with our experiments. The things in black don't mean they're not there, it just means we didn't detect them. But the sequence in blue, right at the far end or the bottom of the protein, is really interesting because that sequence we didn't detect. And the reason it's interesting is because that corresponds to the region of the sequestosome protein that often missing or is altered or is faulty, particularly in these genetic forms of Paget's disease. So once again, the molecular evidence is pointing towards some sort of defect in a protein that we know is intimately involved with Paget's disease. And we were able to use other techniques and other approaches then to validate and confirm our analyses. So this is another way of detecting ancient Paget's protein. Without worrying too much about the detail, if you see a band in one of the lanes, that means it's the presence of the ancient Paget's-associated, the sequestersome protein. So, for example, on the left-hand side, this is an analysis of 2 different bone samples from SK101. So that's, we think, a medieval knight and a crusader from the Norton collection, and William is SK29. He's on the right-hand side. But the fact that you see this banding pattern tells us, or confirms to us, that the ancient Paget's disease, or the ancient Paget's protein, sorry, has been preserved. But the fact that it doesn't look like this lane on the left-hand side, this is what we call our positive control. So this is what the sequestersome protein, the sequestersome protein today looks like. So it confirms the ancient protein is there, but it also supports the idea that it's different in some way. We in total looked at 7 individuals with Paget's-like symptoms, and we could detect the ancient protein in 4 of them. We never detected the sequestersome protein in individuals in the collection who didn't have the symptoms of Paget's disease or the changes associated with Paget's disease. But finally, on the right-hand side is just something that I want to flag up, that we also detected this ancient, probably faulty sequestersome protein, not just in bone, but in teeth. And that might sound strange. Why would we look at teeth in the first place? The reason we analysed teeth was because museum collections are obviously reasonably reluctant to allow us to sample and to destroy museum samples. Teeth are very happy for us to work with because it's easy to make a model of a tooth and to replace it in a display. And so by chance we analysed teeth from these ancient skeletons, and certainly in at least one, I can't remember, possibly 2 of the teeth, we found evidence of the ancient sequestersome protein. And this is of particular interest to me and to us, because Paget's disease isn't associated with changes in dentition. Teeth are not bones, essentially, but something clearly is going on in these ancient teeth where the Paget's protein is present in high levels and also seems to be different to the contemporary protein as well. So this really just adds to the mysteries, I think, of Norton Priory. So almost done, I think. A few people, I think, have mentioned today this idea of genetic factors in Paget's and the fact that very rare, a small number of cases of Paget's are caused by a familial. They run in families, so they can be inherited. It's not common at all. But an obvious explanation of the ancient Paget's disease, or the cluster of ancient Paget's disease at Norton Priory, was that perhaps the individuals were all related. That would seem logical. That also would be supported by the fact they're buried at the same site. Some of the archaeological records suggested that. And also some of them were buried in family chapels. Now, we collaborated with Professor Turi King, and she was the geneticist who actually did the DNA sequencing at the University of Leicester to confirm the identity of Richard III's remains. And she was able to extract ancient DNA from some of our samples with this unusual ancient Paget's disease, and to check that sequestersome gene, which is common, which carries the mutations or the faults in familial Paget's disease, and certainly look for the most common mutations associated with familial Paget's disease. And again, just to add to the mystery, there was no evidence. So this is now an 800-year-old DNA sequence, but there's no evidence that the mutations that cause familial Paget's disease today were present in any of these individuals. So the Paget's associated protein is faulty in some way, is different in some way, but their genes don't appear to be. And so I don't really have an answer for why that is at this stage. So, final slide. Research often raises as many questions as it answers, and we are still really amazed that this work was successful, but we still have many questions to get to grips with. I'm pretty sure this was an ancient form of Paget's disease. The fact that that's a sequestosome protein, of all the bars in all the world, we detected the Paget's biomarker, tells us that this unusual bone disease was a medieval form of Paget's at Norton Priory. An obvious question will be, well, have we just completely overlooked endemic Paget's disease in medieval times? Was Paget's just really, really common, but not necessarily a problem because of different or lack of complications? And maybe you'll Google it, but certainly the Daily Mail interpretation was that something like half the population died of Paget's disease in medieval times. It's not true. I don't think we have overlooked ancient Paget's disease, because actually other archaeological collections have been examined, and it doesn't look like this 20 or 30% incidence was present in other parts of the country. There may be more ancient cases in the northwest of England, which actually historically and still today is a hot spot for Paget's disease, but there's something very, very unusual about Norton Priory and that collection of individuals. And I visit Norton Priory regularly. I was there last week, and I'll be there in a couple of other weeks, and I always joke, don't drink the water, but I do take a flask with me, but I don't know, actually. I'm being flippant here. But clearly there's something about Norton Priory or that location. There's some factor there that exaggerated the Paget's disease, exaggerated the symptoms. And this comes back to the idea of needing to know more about what the factors are, the risk factors that control presentation of Paget's disease. And now our attention with the museum and with other collaborators is moving towards looking more closely at environmental factors to try and identify what they might be. Is there something in the soil? Is there something that's under our noses almost that could be kind of the missing link that explained why Paget's was so unusual? I mentioned that Paget's has changed in the last 10 to 20 years as well. So not only was it very different back in medieval times at Norton Priory, but actually across the world, or certainly in some countries, the incidence of Paget's has dropped rapidly. The extent also seems to be declining. Monoostotic is becoming more common, and the age at presentation seems to be increasing, which is associated with reduced severity. So that's all good news for Paget's disease, that the condition seems to be going away. But we don't know why. And I think having an understanding of the factors that control the kind of presentation of Paget's symptoms is really important, because if we can understand what those factors are, then of course we can mitigate those factors, we can make very simple interventions maybe to avoid those factors. And I think in that regard, moving forward, then the future looks very bright if we can learn from this, from what history is telling us, and if we can apply that to Paget's disease today for the benefit of our members and for those affected. I mentioned that the Paget's Association has ambitious plans to use some of the assets we have, some of the money we have to fund research programs. So we fund research projects, we've touched on the student research bursaries. We've also very recently recognised that there's a need to support educational research as well in many guises. And that is as simple as raising awareness of Paget's disease, but could also include pure educational projects. And so we've launched an educational research award scheme. So again, small scale, but hopefully will make a bit of a difference. And this story is actually the first that's going to be investigated and told, hopefully through an education award that's recently been awarded to a student in Nottingham. So the award will be made in a moment by Professor Grayson Ditchfield, who is going to say a few words, I think, and just introduce, I'll say a few words about the background to this award. So our educational awards are now live, or will be going live very soon with our next round of grant funding. And in this case, the educational award is really trying to capture lots of the media attention that's come from this story, and to try and, I think, give it longevity. So we raise the profile of Paget's disease. People out there know about Paget's disease, they know about us, they know about the association, they know about you guys. We want to generate resources as well, displays, to spread the word, essentially. So the Educational Award is going to, as I said, be presented by Professor Grayson Ditchfield, and this award is going to be in the name of his late mum, Winifred. And so Professor Ditchfield and his mother have been great supporters of the Paget's Association over the years, really tremendous, worked again tremendously hard with us, and we really do appreciate that. So we would like to name the Educational Award after your mum, and Grayson, I think, will just say a few words, and then we will make the presentation to Elizabeth Radley, who's a student here in Nottingham, but we're not going to put Lizzie on the spot, so we'll put Grayson on the spot briefly as well. So thank you.
Thank you. Well, the programme is entirely correct in predicting that I'll only say a few words, to assure you. It's very kind of you to invite me here, and I'm greatly honoured to be present. And all the more so because this award has been named after my mother, Winifred Ditchfield. I should add that my mother was a Paget's disease patient, that she was a member of the association from very early in its development, and that she was very well informed about bone conditions because she had worked at Manchester Royal Infirmary as the secretary to one of the most distinguished orthopaedic surgeons of his generation, Sir Harry Platt. And the entry for Sir Harry Platt in the Oxford Dictionary of National Biography is well worth reading. I should say that my own credentials for talking intelligently about Paget's disease are barely zero. But, on the other hand, as a historian, I find it fascinating to see how historical research has been transformed by technology, and the research at Norton Priory, about which we've been hearing today, is a superb example of precisely that development. And the more this research is known about, and the more that Paget's disease is understood in its widest context, including its historical context, then I think the better for all concerned. And so it's a pleasure to be able to present the Educational Award to Elizabeth. Well, congratulations, Elizabeth, and we wish you every success in your work, and we look forward to reading more about it in the future.